All right, hello, and thank you for joining us behind the science. Here we will connect you with Eurofins network of testing labs and more broadly with the science of food testing in a way you may not have experienced in the past, from the viewpoint of the scientists themselves. This conversation aims to zoom into a single anecdote of the Eurofins story through the personal accounts of our guests. If the topic raises a question for you, feel free to reach out using the questions link. We will not address your questions here. However, we will follow up with you afterwards. So to set the stage for today's episode, our guest is Dr. David Legan, Director of Science at Eurofins Microbiology Laboratories. I am Scott Mushakian, playing the role of moderator, and our host is the Chief Scientific Officer, Dr. Douglas Marshall. Doug, I'm going to go ahead and hand it over to you to start the conversation.
Thanks, Scott. Really appreciate everyone joining us for our second episode today. We appreciate all the feedback you gave us on the first round, and we've done some improvements, we hope. But please keep those suggestions coming. I'm incredibly happy to have David with us today. David's a very close colleague. He's a phenomenal food microbiologist. So kind of the technical stuff we're going to go over today is really related to what David brings to the table, both to the profession, but also to our customers. And so hopefully we'll have an interesting conversation about all the dirty little details that a food microbiology testing laboratory has to deal with, both in terms of the methods as well as kind of the customer expectations on how the laboratories are really designed and be able to use the appropriate methods to deliver data that's meaningful for our customers. So David, welcome. I appreciate your being here with us today. If you wouldn't mind, if you wouldn't kind of like to share a little bit about your background and maybe how you got to this point within the Eurofins network.
Thank you, Doug. I hope I'll be able to live up to the introduction. And this part's a long story, so I'll try and keep it as short as I can. You can tell from the funny accent, I am not originally from the US. I graduated out of the UK with a bachelor's degree in applied biology with a microbiology specialization, took a PhD in food technology, and then I worked for 12 years in a small bakery institute doing baking research and working very closely with UK flour millers and bakers. That institute later became part of Camden BRI, which to some people will be familiar. It's very well known internationally. During the time I was there, we did a lot of government contract work. And as a result of that, I got very involved in pathogen modeling. It was sort of the flavor of the decade with the USDA and the Australian government, the French government, the British government, all very much active in funding modeling programs. And the work we did there is actually available now in the models in Combase that's accessible online for free. After that 12-year period, I had a rather strange and unusual collection of skills, and I was headhunted actually into Nabisco Research in East Hanover, New Jersey, to bring together bakery microbiology and modeling and do some preservation system development, which I did there for 3 years. And then, the usual sort of corporate acquisition activity went on, and Nabisco became part of Kraft Foods, as a result of which I moved out to the Glenview R&D Center just north of Chicago. And I continued to work there on preservation technology, really. Over time, that work led the company to launch a range of high-pressure, pasteurized, ready-to-eat natural meats, and that's kind of a mouthful, but I try and keep it all together. Also worked on applications of microwave sterilization technology in partnership with Washington State University, ran a discovery program looking at synergistic actions of natural antimicrobials, which may in fact be very topical today as we get into the discussion of natural everything. There are many very effective natural antimicrobials, and a lot of them taste terrible. So, if you want to use them in a food product, the question is, can you find synergistic interactions that allow you to make those practically effective? So we actually used a pharma-style high-throughput technology approach. Towards the end of the time at Kraft, I was running research and development groups supporting basically preservation systems across the entirety of the Kraft range of US brands. So lots and lots of interesting stuff going on. But again, corporate activity happens, and Kraft was acquired by private equity. Their thinking was they didn't need anything to do with this research stuff. It was just a cost on the balance sheet. So many of us found ourselves looking for another opportunity. And I was actually very fortunate that there was this small microbiology lab in Madison, Wisconsin, Covance Food Solutions, and they were looking for somebody that knew a little bit about microbiology to help out with some of their method development and method validation questions. So that was about 120 miles from my home. It was great. It built on what I knew. It was conveniently accessible. And it was actually a really fun time for 2 years working on method validation and development. And then again, corporate stuff. And Covance Food Solutions was acquired by Eurofins. But this time, fortunately, it worked out, and I found my role broadened out from the two Midwestern Covance Food Solutions labs to working across the entirety of the Eurofins microbiology network, which at the time I think was about 17 labs and is now up to 25 and growing. So, life just gets more and more interesting. I think one of the things that I wanted to kind of highlight, I know that was a long background, I've actually been on both sides of the lab door. I started out my career, as a service provider inside the lab. I spent about 20 years as a customer outside the lab, and I've spent the last 9 years back behind the lab door as a service provider. So, I get that there are things that are just frustrating on both sides. And we do what we can to try and minimize those frustrations. But on the other hand, life and nothing's perfect. So we do what we can. Now, I know that was probably longer than you really wanted, but it did summarize a 40-year career into about 5 minutes. So it's a pretty good compression ratio.
Thank you very much, David. You have a phenomenal background, and our customers really benefit from the breadth and depth of your experiences. So you mentioned a little bit about working in the Covance laboratories, and just curious, you also mentioned now you're supporting all the food microlabs. Can you just briefly describe the test portfolio that you support?
Yeah. So it's really, it has two components to it. Probably the largest component by volume now is the Eurofins microbiology network that is not the Madison lab. And that really is the entire portfolio of sort of standard microbiology tests: Salmonella, Listeria species, Listeria monocytogenes, E. coli, rapid screening platforms of various kinds, and then the range of indicator tests that people would or might want to do, including coliforms, E. coli, lactic acid bacteria. So both the high-tech and the traditional cultural microbiology, detection methods, enumeration methods. And I don't do this alone, Doug, as you know. We have a small, geographically dispersed and highly skilled team that we loosely call research and development. I want to credit my colleagues, Dr. Chris Crow and Erica Miller. Between us, we have actually a very complementary skill set spanning, the food industry, the diagnostic industry, lab operations. And there's not very much that we can't at least turn our hands to and mostly we managed to come up with solutions. Then the other component remains connected with some rather high-tech stuff in Madison, Wisconsin that we developed alongside the probiotic test portfolio. So there's some USP testing, there's some sequencing, there's some probiotic testing. And here again, I want to basically credit Andrzej Benkowski, who is our probiotic guru. I think you should have him on one of these sessions at some point in the future. He's a great storyteller and he knows a lot about a very unique part of microbiology.
Agreed. So one of the things that this small R&D group does, and occasionally we have customers that ask a question like this. So I just saw some new technology that they claim they can do a pathogen test, let's say in 2 or 3 hours. And part of your role is to kind of vet these technologies and ask questions about are these claims real, and are there commercial opportunities in our laboratories? Do you have any kind of thoughts about all these new technologies that seem to pop up every single year? The vast majority of these don't succeed in the marketplace, and so maybe you just have some thoughts about why that might be.
I may have some thoughts, Doug, and I will say they're not unique to microbiological test methods. I found during my time in the food industry that many companies have the best ingredient under the sun, right? Tasteless, calorie-free, will solve all your preservation needs, will solve all your color needs. They've sold it to the CEO, and now it comes to the technical organization who have to make it work. There are a lot of people that are, fundamentally out there trying to sell their thing, and more power to them, right? That's their business. Our thing is to figure out, can we make an application from these technologies? And we put quite a lot of time into this sort of technology scanning, technology scouting. We have conversations with these companies at least a couple of times a month, and you're on some of those calls. You go to some of those visits. Most of the technologies have something going for them. I think the reason that they don't necessarily make as much progress as their salespeople would like is they tend to be very expensive. They tend to struggle to deliver on the time promise. So yes, indeed, maybe you can get a 3-hour pathogen result because you're starting at the back end of the enrichment culture that you prepared in the lab as a demonstration. You're not starting with the sample that comes through the lab door that has to be weighed, that has to be enriched and prepared for detection. We've adopted some of these new technologies, and we'll probably talk about that later. In some cases, they get adopted in a specific niche where they provide a benefit that is large enough that you can afford to pay the extra cost that they bring, or you can afford to, stand the extra labor, or you can train somebody specially. I think it's been my experience that there are very few magic solutions that will make all of our problems go away. That's a good thing, because if there were, then neither you nor I would have a job.
Fair enough. Kind of back to the old saying, let the buyer beware. If it sounds too good to be true, you need to be doing a lot more digging into the technology and the supplier to make sure that it is real. All right, thank you for that. So Eurofins Microbiology has a very large portfolio of tests, and you spend a lot of your time working with our laboratories as a technical expert. Um, I'll just kind of pose this question to you. If you could pick one test that is the most difficult to perform at the bench, could you kind of focus on why that test is what in your mind the most difficult to perform?
Yeah, Doug, I find it hard to play by the rules sometimes, so I'm going to give you three examples.
Fair enough.
They're difficult because they're complex. So the first example I'm going to give you is the sequencing work that gets done in medicine. You know, this technology is kind of unbelievable, and if you think back, I'm not quite sure now how long ago the Human Genome Project. It was $5 billion and about 5 years' work to sequence one genome. Now we don't go quite that far, but we can sequence a bacterial genome in about a day for a few $100s these days. That's how sophisticated the technology is. But it does mean it's very complex to operate. Lots of very fiddly steps, very small volumes, very precise handling, and a rather expensive machine that you can break easily if you do something wrong. So what we have is a couple of very skilled analysts that do this all the time, and they turn out great results. And they probably would tell you that it's not the most difficult test in the portfolio, but that's because they do it every day. And I think there are only really two people in the network that I would trust to run that machine in that way. The other one is actually one of the new technologies that we've adopted that leads to this more rapid result. We have a pathotrack system out in one of our produce labs. And basically that allows us to do a very short enrichment of a large portion of lettuce or other leafy greens, then run that large volume of enrichment under pressure through a filter to concentrate it, and then take that sample into a PCR detection. That allows us to get very fast results. The system is expensive in terms of consumables, pays for itself because of the volumes that we can put through it. But you're dealing with a pressure system, lots of connections, lots of steps, and you don't want to get that wrong if you have a pathogen enrichment. So again, we have analysts that are well trained, quite skilled, but we don't run that in all the labs in the network. And the last one is actually a traditional method. It's Clostridium perfringens confirmation. That's complicated because of the anaerobic requirements. So you have a number of different media, a number of different steps. All of those have to be de-aerated. You have to keep oxygen away. You have to incubate in the absence of oxygen. So there's nothing actually that difficult from a traditional microbiology point of view about it, except keeping the oxygen away. So there you go. Two new and one old method.
I appreciate that background. My own personal inclination is just a traditional plate count method seems to be really challenging, primarily because you and I both have taught, a plate count method, and you have to have a lot of due diligence at the bench to make sure you label everything correctly, to make sure you get your transfer volumes done, and then make sure you know how to count colonies. So that one, just for your information, that would be mine. Probably the oldest test in food microbiology, I think, is still the most difficult.
Yeah, I think that's a fair point of view. And certainly that's at the level of, we have lots and lots and lots of people doing that test, right? So they are jacks of all trades, not specialists in some narrow field.
All right. So when we present a test catalog to our customers, we may have, if someone just says, I want an E. coli test, for example, and our business development staff or our customer service staff, they will prepare a quotation for the customer. And one of the challenges is, picking the right test for the
Hmm.
customer. And we rely pretty heavily on customer feedback in terms of what it is they want and why. And so, if you could just think about our portfolio and put yourself in the buyer's perspective, um, can you think of maybe one or two tests that is just really challenging for a customer to wrap their heads around about? I want this test. So maybe that question makes sense to you, maybe not, but I'll put it in your lap to
Yeah, no,
contemplate.
it does make sense, Doug. And I think the space where I have interesting conversations and it gets complex is in the probiotic space. And you'd think, I want a probiotic test. Well, we have 1,500 different test codes because we have to consider the species, in some cases, the strain. Many of the manufacturers have their own bespoke methods that could be available only to their customers. We have internal methods that do similar things that we've validated. And depending on the application for the probiotic, you could have a very wide range of expected concentrations. So you're dealing with multiple inputs on the what am I testing for, and multiple questions about how much do I expect, how much do I need to dilute to get a number, and then am I doing this by a traditional plate count method? Am I doing this by a high-tech method like flow cytometry? If I'm doing flow cytometry, do I just want a count? Do I want a differential count of spores, of live, injured, and dead? So typically what we do there is engage with Dave Roth, the business development director, who is really good at talking customers through what their needs are. And then we'll provide them, if you like, a short test list or list of test codes that they can choose from. And that steers them into the direction of what they need. Now, they can always come back at any point in the future and say, my needs have changed. I want to add some other kind of test. I need to add a different dilution, and we'll rebuild that list for them. But in the short term, we provide them a cut down list and try to help them to not pick the wrong thing.
Great example. Thanks, David. Now then, once we execute a method and we get the analytical results ready for a certificate of analysis, and we send that analytical report off to a customer, many of our customers aren't microbiologists. Maybe they're new to the industry that they're in, and they really struggle to interpret what these analytical results actually mean. Can you give me maybe an example of a test where we have a lot of customer interaction to help them interpret an analytical result?
Yeah, again, there's a few. One that comes up moderately frequently is when we're doing USP testing, United States Pharmacopoeia testing. That's rather different than the run-of-the-mill food testing that we do. Every single USP test gets what we call a suitability or preparatory test the first time that we encounter a matrix. And essentially what that is, it's a little challenge test where we spike a sample with the target organism, run the test, then look to see, if it's a presence absence test, did we detect that organism? If it's an enumeration test, did we get a count that's within a specified range around what we put in? And if we do, that's great. That test reports as passed, and we go on to do the regular testing on the sample. If we don't, that test reports as failed. And failed is, a difficult thing to see on an analytical report. So, we get into explaining that It doesn't mean that the product failed. It means that the method failed and it is showing us not suitable for this matrix in its current variation. Now, there's a number of options that we have for neutralizing some of that inhibitory activity that may be associated with the product. So we go through a number of steps of dilutions, different inhibitors, and if we get eventually to a variation where we can get that test to pass, all well and good, we go on to the testing. If at the end of the sequence we're still failing, then essentially we provide a USP blessed form of words that says, something along the lines of this matrix is inhibitory and it's not very likely that you'll find the organism that you're concerned about in it. That one takes some time.
Okay. So, from a customer perspective, I would venture a guess that the vast majority of customers really have no clue that this is the behind-the-scenes activity that we have to do with a specimen that we're not familiar with, or with a specimen target microbe combination that either there isn't a published reference method that's been validated for that matrix type and for that target microorganism. And, we have thousands and thousands of customers bombarding us with tens of thousands of different matrix samples on a daily basis. So the complexity of what happens at the bench gets very severe fairly quickly. Now, that's a little bit different if we have a relationship with a customer, we're familiar with the matrix, familiar with the method. Those are the good days, but we oftentimes have the challenging days. So having that background example, I think, is hopefully the folks watching this will appreciate that this is not an easy task that we do at the bench.
Right. On the general microbiology side, Doug, the method we run a whole suite of methods that have formal validations for the most part, and we have internal procedures for matrix verification. So once we step outside the realm of the method validation, we certainly have procedures on the general lab side of the business to verify matrices, and we can either do this directly when we're onboarding a customer. We certainly would do it if any question arises about the ability of a test to detect with a matrix. We'll sometimes do it on a precautionary basis. We get something come through the door and we can look at it, or from the label say, I'm really not sure about this. I strongly recommend that you would do some verification before we do testing. But it's not so built into the process as it is on the USP side.
Mm-hmm. Okay. So, David, you mentioned a little bit about sequence methods. You mentioned a little bit about some flow cytometric methods for probiotics. Can you give me an example of another new test that Eurofins Microbiology has onboarded recently, and why that might be of value to our customers?
Yeah, there's a few, Doug, actually, when you look over the last few months and look forward. One of the things that we've done with sequencing, so it's not the sequencing is the new test, but we've been working with our colleagues in France and partner Eurofins labs around the world. Some of our multinational customers have asked us for the ability to track pathogen strains around their networks and see if they've ever encountered them before. And so we now have a very standardized protocol that any of our internal labs connected with this procedure can run, send the sequencing files electronically back to the bioinformatics lab in France. They run the informatics and can say, yes, customer X, indeed this was a salmonella, and it's this many SNPs different from the salmonella that you found 6 months ago in another country, or it's completely different. And then they can offer a customized database. So that's a way in which we're really moving the technology application. At the wet lab side, we've recently brought on board a more rapid modified salmonella confirmation method. We're using immuno-labeled magnetic beads to go into an enrichment, pull out salmonella in a concentrated fashion, and then go into a PCR detection. And that saves several days over the classical sequential culturing and subculturing and picking things that look right at one stage and moving them forward to the next stage and see if they still look right. Depending on the time of day we get a screening detection from a rapid method, we may get a result from this modified immunomagnetic method by the end of the same day. If the sample comes out late in the day, it's more likely going to be the next day, but much quicker than the cultural method. We've also brought on board a PCR-based salmonella serology method. So running 27 different, or tests for 27 different genetic markers in a single multiplexed PCR assay that can identify 59 of the most common salmonella serovars on the officially validated method, and we have some internal validations that extend that. If by any chance an isolate does not fit within the validated scope, we can always fall back to the traditional serology. But here again, this is a more objective, quicker method, and customers are finding it quite beneficial.
Great. It's nice to see that our laboratory services don't stand still, always looking for something newer and better.
We never stand still, Doug.
Absolutely. I'd like to turn now and maybe spend a few minutes on, you do a lot of customer interaction. Our customers really value your interpretation and your recommendations on testing. And I don't know if you have maybe a couple of frequently asked questions that cross your desk on a maybe a more regular basis than some of the odds and ends that you might be presented with.
Yeah, it'd be fairer to say that there are categories of questions.
Mm-hmm.
And so, one category, we've talked a lot already about screening methods and confirmation methods, and maybe the biggest single category of questions is around what's the difference between a screening method and a confirmation method, between a presumptive result and a confirmed result? What's the significance of a presumptive result that doesn't confirm? So there's a whole stack of questions, and they're parallel whether you're dealing with Listeria or E. coli or Salmonella. So, you see how the permutations of the question multiplies rapidly, but it comes down to, a few relatively basic things. So one place where this plays out is, you and I remember a time when there were no rapid screening methods. And as PCR and immuno methods came on board about 30-ish years ago, they were validated for sure, but they didn't have much of a track record. And so it became conventional to say, if you run a rapid screening method, you don't actually have a positive or a detected result. You have a presumptively positive result, because we're not quite sure. And then we'll take that presumptively positive result back into a traditional method and get you a definitive answer. So, we talk about presumptives for short and confirmed from the traditional flavor, the traditional cultural method. Now, the screening methods don't necessarily detect live cells. They detect components of cells. So a fragment of DNA or a cell surface antigen, which means that if there was a large population of that target present at some point in the past and it's now dead, you may detect it by the screening method, but you may not detect it. Well, you won't detect it; it's dead by the cultural method. And we have some ways to mitigate some of those things. We can digest or inhibit the DNA from dead cells. That helps a lot. Another thing that happens is, nothing happens in a vacuum, and bacteria have lots and lots and lots of relatives. And some of them grow in the same kinds of spaces and survive the same kinds of inhibitors that we use in selective methods. So, as an example, there are 21 species of Listeria now. And the one that we're most concerned about is monocytogenes, that's the pathogen. But they all grow in basically the same spaces. They all like low temperatures, they all like wet environments, and they all tolerate the same antibiotics. So if you're looking for Listeria monocytogenes and there's a large background of other Listeria present, you could simply overwhelm the monocytogenes signal when you go to the cultural method. And you think, well, okay, let me dilute the sample so that I don't have this enormous population of the non-mono on the plate. Well, yeah, but you've also diluted the population of mono, and maybe it's not there at all on the plate. So, that's another place where it gets to be complicated. And the final place where it gets to be complicated is, we're really pushing at the limits of some of these technologies. And with the best will in the world, every test method has a true false positive, a true false negative rate. The developers work to make those very small, but it's hard to get away from. And in the clinical space, people are very, very aware of this situation, right? And decisions get made about, should I run a screening test on a patient with no symptoms? Well, the occasional false positive may send you off for all kinds of horrible follow-up confirmatory tests that cost you a lot of money. In the food space, we don't really tend to talk about this. There's a sort of assumption that methods are perfect. There's no way round the fact that we need to be doing lots of testing to make sure that we don't have pathogens in a food facility. But even if you have a 1 in 1,000 false positive rate, when your incidence of pathogens in your screening population is small, and we're seeing less than 1% of samples come back with a presumptive at all, then, you're looking at kind of naturally 10% of those screening results will not confirm. And there's really no way around that. And it's very frustrating for all parties. And we're now so far down the road into screening that we've forgotten how marvelous it was when you could get a result in a day or 2 days, not 5 days or 7 days. Now it's really annoying when you have to wait an extra 5 days to confirm the result. So just some context and perspective on that. I know it's frustrating, but most of the time you get a result really quickly and can release that negative sample. I'm sure you also, like me, get lots of questions about how to interpret, a strange result. We've sort of touched on that already. I get lots of questions on what limit should I put on name your test, aerobic plate count on green tea. And there are ways that you can actually set that relates to the matrix itself, the process that's used. But often the request for that answer is, I need it yesterday. And so then we go look in the literature, see what guidelines have been produced by governments, see what some retailers are saying as their acceptable limits, see what you can find in the research literature as typical for the product, and then go back to the customer and say, well, look, here's what I've been able to find. Maybe this is a good starting point, but then over time, maybe it would be a good idea to build your own number based on what you know that the process can do. And the other thing that we get quite a lot of, well, that's sort of out of context. The thing that I see fairly frequently is questions around, my product is spoiling, help. And that manifests in many different ways. Based on the symptoms of that spoilage, we can often make guesses about what might be the underlying cause. And then if this has come from a lab, these often come through a lab, then the lab can do some additional testing to figure out what the direct cause of the spoilage is. And then we can bring in our colleagues out of the Advanced Microbiology Group or the National Food Lab. They can bring to bear expertise on process capability, process validation, preservation systems, product formulation. And so we can not only diagnose what's the immediate cause, but we can often help to make sure it doesn't happen again. I think that's probably a long enough list of strange questions for you.
That is a lot to unpack, and yes, I'm fully aware that our FAQ list is much longer than these examples. So, David, you and I have been in this business for a long time. Some of the tests that we perform far exceed our respective ages put together, and the methods haven't changed. Just a standard plate count method, for example, is quite old, and really few improvements have occurred with that method over time. So I'm just kind of curious, if you put your futurist cap on, what do you think the future looks like for food microbiology testing? Very open-ended, I know, but that's the point of this conversation.
I think the good news, not necessarily for you and me, Doug, but the good news for our younger colleagues is I don't think it's ever going away. Bacteria ruled the world long before we were ever present, and I think bacteria will rule the world long after we're gone. So, job security for microbiologists, I think, is assured for as long as there are microbiologists. Beyond that, I'm not the first to say it's hard to make predictions, especially about the future. So what we tend to do then is extrapolate. One of the interesting things that I've seen happen over the last few years is the way that sequencing applications have developed. So when we first started dabbling with this, me and Madison and you, I think, with the lab in New Berlin, the food industry in the US was basically absolutely opposed to anything that might give a pathogen sequence because the consequences were potentially terrifying and nobody knew what it meant. And I think we were very fortunate in Madison that we had this big probiotic client base. They didn't have a pathogen concern, but they were very interested to know, what's in my blend? Is it what I think it is? What did my supplier send me? Is it what I ordered? So they were very willing to explore sequencing and saw value in it, and that let us build some capability. And that capability is now available. And I think what we've also seen is that the multinational companies have built great experience of sequencing applications in Europe. The regulatory framework is quite different. They don't have the same, perhaps, fear that they did here about what might be the consequences. And they've got very comfortable with the notion of, I find a pathogen, I sequence a pathogen, now I can track a pathogen and make sure it's gone from my facility. I can know where it came from. They're bringing that confidence globally. And so I do think that now over the next few years, we'll see that confidence begin to build out across the US food industry. At least I hope that it will, because this is a really very powerful tool that can help people with their food safety programs. The other thing that, the question is always, can I get this result faster? We get that question every week. As you mentioned, there are lots of people that are selling a faster result until you go and investigate the details. So I don't think that we're going to see the leap forward that we saw 30 years ago from culture into, these molecular methods. I do think that we will continue to see niche progress where we can get to faster results in specific circumstances for specific organisms, for specific matrices. That developing those approaches takes a chunk of our R&D time, and I think we'll continue to make progress, but it will be more limited than it was in the past. And I think it's worth keeping in mind that what we're trying to do with pathogen detection is very, very, very difficult. The methods have been around for 100 and some odd years in many cases, so we kind of assume that it's not hard. One of my colleagues got me to do a calculation a few years ago to figure out how hard this is. If you think of, a Monet haystack, 18-foot diameter, 18-foot high, and you put a 3-inch needle in the middle of that haystack and try to find it, that's kind of seen as the ultimate in difficulty. Well, if you do the calculation, the concentration of one bacterial cell in a 375 gram sample of food is about 170,000 times lower than the concentration of a needle in a haystack. So, we're really pushing at things that are very, very difficult to do.
Very good. Well, David, I think our time is coming to a close. I'll leave us here with one final question, and that is, if you don't mind sharing with the audience, what is the most rewarding part of your role in Eurofins, or what's the favorite aspect you like working in the Eurofins environs? I think that would be helpful just to be able to demonstrate why we have people of your stature and your abilities working in our company.
Yeah, I'm sure your world too is like mine. Every day is different, and you don't know necessarily from day to day what's going to come through the door or over the phone or through the internet. Probably the one thing you do know is that you've got more questions on a Friday than any other day, because that's when the world falls apart. But certainly, working with our customers on a whole bunch of issues is endlessly fascinating. And one of the nice things about having been around for a long time is you start to see patterns repeating, and you start to recognize in questions that they're addressing maybe a surface phenomenon and not the underlying issue. So it's quite satisfying to be able to have a conversation, ask a load of questions, and maybe move a customer to a place of realizing that, yeah, this thing I thought was a problem, it is a problem, but it's caused by something else, and we can fix the something else. And one of the really neat things about Eurofins is the just astonishing amount of capability and capacity that we have within the group. And we can bring all of that to bear on a customer issue as and when it's appropriate. So, never a dull moment, and the ability to help people solve their problems. There's not a lot to not like about that. I think the second thing that I find it really gratifying is the ability to work with the younger folks in our network. We have a lot of very smart, very dedicated people working for Eurofins Microbiology that want to do good in the world. And the ability to maybe help them think through Do they want management careers? Do they want technical careers? If they want technical careers, those are the people I tend to, you know, work with and mentor more. How do they build their network? How do they build their skills? These people are the future leaders of Eurofins microbiology and Eurofins. They're going to step into our shoes when we're gone. So, again, very, very satisfying, very gratifying to be able to build people and pay back some of the advice I was given many years ago. And the other thing I've been very privileged to do is work with colleagues in other lab networks, our competitors actually, folks in the diagnostic testing development world, in food companies, and in government. And over the past 5 or 6 years, we've put out a number of kind of educational articles through Food Protection Trends, talking about some of these more nuanced aspects of testing. Some of the things to think about with validation, with method selection, and so on. Those articles have been very widely read, have been recognized as the most read in a number of different years that they've come out. So they're doing some good. So I think at the end of the day, it's this job provides endless opportunities to try and make the world better. And it probably sounds very idealistic, but there you go. That's kind of why I got into food in the first place.
Well, David, it is a privilege in my career to be able to have a colleague like you to work with, and I really do appreciate you sharing your background and your experiences, because that's what this webinar series is all about. It's the individuals we have in our company that bring out the best in what we do, and hopefully make us the best in the industry. So I appreciate you sharing your thoughts with us today. And Scott, we will close this one out, and I look forward to the next episode, and I wish everyone has a pleasant day. Thank you.
Thank you, Doug. Thank you, Scott.